I'm going to talk about our work on setting up a framework for the comparative biochemistry of cellulases. So we want to set up something that we can compare the activity or performance of cellulases. And I'm going to have a particular focus on how we can use such a framework for predicting kinetic parameters from the sequence of these enzymes. This is a long-standing collaboration that my group at BTU just north of Copenhagen we've had with Novozymes, and very recently actually Novozymes, or a year and a half ago, I think Novozymes moved part of their research facilities to an address right across the street from us. So now we're opposite neighbors, and that obviously further has stimulated our collaboration. So the problem is with all this that we want to characterize these enzymes, but the substrate is solid, and that's kind of unusual. So that sets up some extra challenges. And of course, when we want to characterize these enzymes, we're interested in parameters such as those here: specificity, regulation of the enzyme, and their kinetics. And I think, put a little bit boldly, I think it's fair to say that the two other parameters, specificity and regulation, we can pretty much assess by methods and approaches that are similar to the ones we use for normal enzymes in solution. But when it comes to kinetics, which is what I'm going to talk about today, there are some additional challenges. So it's actually very tedious to describe the kinetics of a cellulase in a systematic way, and in a way so that the parameters you get out are rooted in some kind of a physically meaningful model or theory. So since it's tedious, you might start by asking yourself, why do you want to do this? Why is it worthwhile to kinetically characterize these enzymes? And one of the best answers to this question that I've been able to find, it's very, I think, very eloquent and concise, is from this work by Blickard et al., who some years ago said that kinetics provides the experimental link between structure and function. So I think you can't say it much more precisely. And actually, these workers were interested in not in enzymes, but in inorganic catalysts. So I think that just proves that serves to prove that this is a very general and overarching principle that we can use in all kind of catalysis. So what I'm going to talk about today is how can we establish such a link, and to sort of zoom in on the challenges. Let me outline some of the problems that has been discussed a lot over the years. Why is it tedious to describe enzymes kinetically when they act on an insoluble substrate? Well, perhaps the most obvious reason is that there's an extra level of complication because the kinetics depends on the physical properties of the substrate. So parameters such as pores or particle size, length of polymers, crystallinity, etc., we all know that has a very distinctive influence on the kinetics of these enzymes. But it's not something we have means to describe in conventional kinetics, because for a soluble system, those things don't occur. They are not problems, so therefore they haven't been addressed. Perhaps well, another very fundamental problem, let's see if I can get this to work here, is that the pivot of all kinetics, as we know, is the law of mass action, or so-called mass action kinetics. And that's exemplified here. So, for instance, the rate by which an enzyme and substrate form a complex is some constant times the product of the free concentrations. And that is, of course, the molar concentrations, because that has to do with the collision frequency and all that. But that becomes a little bit questionable, and it's not unclear what that means. What is the molar concentration of an insoluble substrate? Of course, there is no such thing. So therefore, by definition, mass action kinetics is not directly applicable. And that's been discussed extensively in the literature, whether that's a showstopper or not for this kind of work. Very often you just neglect that and insert the substrate concentration here in terms of a mass load, so it's a grams per liter or whatever. And then it works nicely, and I'll return to that in a minute. But there's an associated problem with that, because then the parameters that you get comes out in odd units. You will get units that are not conventional units in chemical kinetics, and they're hard to compare to other enzymes, and you can't compare them across studies made in different labs. So the comparative aspect is still challenged. Finally, there's a lot of discussion about whether you can have a steady state for these kind of enzymes, because as you know, most handy kinetic descriptions are based on a steady state or quasi-steady state assumption. So these are the problems we're facing, and how can we deal with that? Well, a key question is, should we try to adapt the well-known enzyme kinetic theory. So in other words, she would take a look at the Michaelis Menten framework and try to adapt it as much as, or as little actually as possible, to enable description of these interfacial enzyme processes, or should we ditch that altogether and try to develop a whole new kinetics based on different principles? And personally, I think that we need to apply both of these approaches. But when it comes to the comparative aspect that I'm going to talk about today, it's very important to have a very simple kinetic framework, because otherwise you drown in data and it becomes too complicated, and the demands for experiments are too high. So you're never going to get enough data to make a real broad comparative analysis. So therefore, we have tried actually both strategies, but one of them has tried to think about what is the blockage of using the Michaelis Menten framework. And I think that the real problem is this view of a stoichiometric description of the reaction. So stoichiometric obviously means that the particles sort of react as they come together, one by one, or two and one, or whatever. So therefore, it doesn't work with a mass-based unit. And therefore, I think the very important step forward to use this is to realize something that was realized actually over 100 years ago by Langmuir and others. Actually, those were at the same time as Michaelis Menten, and they realized that if you convert the surface into some sites, then you're okay. So let's convert the surface of cellulose into some attack sites, or consider some attack sites, and define the substrate in that way. What that means in practice is that we get one additional kinetic parameter, which is the attack site density. So we usually use the symbol gamma for that. That simply means how many sites in 1 gram of cellulose are there at which a given cellulase can combine and make a productive complex. So they can make a catalytically active complex. And of course, this is not a universal constant. It depends on what type of cellulose you have, and it depends on pH and temperature, and it depends on how far progressed it will change through the reaction when you sort of start to modify your cellulose surface. So it's not a constant. It's something you have to measure experimentally under the fixed set of conditions. But that's actually the same as the other kinetic parameters, such as Km, etc. So that's nothing special. But there is one extra parameter, and I think that's crucial. So if you look at the consequence of that, let's consider a cellulose particle. So here's one piece of cellulose that's suspended in water, and we look at the surface, and we enlarge one little piece of this cellulose surface, and then this cartoon representation, we see that it consists of small sites here that are squares in this particular cartoon, and that square defines a place where the cellulase, one given enzyme, can combine and produce a product. Now, if we look at it that way, we can define an apparent molar substrate concentration. Because if we know how many sites, how many places in 1 gram of cellulose can react, and we multiply that by how many grams of cellulose are there in 1 liter, for instance, then we get the number of sites in 1 liter. So that's a apparent molar concentration of the substrate. And that's very useful because, first of all, this is an experimentally measurable parameter, and second of all, it provides a framework to use normal kinetics for these type of systems. And actually, at least that's what that's my postulate, is that if we do that and we set up an experiment so that the number of those sites, as I just mentioned, the molar concentration of these attack sites is a lot larger than the molar concentration of enzyme, then it's fair to assume a quasi-steady state, and we can use a normal Michaelis Menten framework. Of course, this criterion here is exactly the same as the one we use for soluble systems. It just follows from the introduction of an apparent molar concentration of the substrate. So this can be discussed a lot, and I'm not going to do that today, but I'm rather going to show what are the consequences of applying this idea and interpreting cellulase kinetics by simple Michaelis Menten kinetics. So here's an example of how it may look. Here we took 2 enzymes. These are enzymes from Trichoderma ricei, cell 7A and cell 6A. They are the 2 most well-investigated cellulases that we know of. And you see, at the first glance, at least the curves look like normal Michaelis Menten curves. You get an asymptotic approach to a maximum value. So far, so good. Now, and you can also see that the normal Michaelis Menten equation, which is fitted by these lines here, seems to account pretty well for the data. So at least you can account for it for the experiments with a normal, just a conventional Michaelis Menten equation. The parameters that come out of that are the conventional ones. So there's a maximal rate that's obviously defined in the usual way. It's asymptotically approaching this value. So that's Vmax. So if we divide that by the enzyme concentration, we get kcat in the usual way. So that's a maximal specific rate. It's not very high. It's only less than 1 per second, but we'll return to that in a minute. But right now, it's just important to emphasize that it has a normal meaning, like kcat has in a soluble system. It's also important to emphasize that kcat does not mean the rate of the catalytic cleavage of the bond. Now, the actual catalysis, the chemical transitions that happen when you hydrolyze the glycosidic bond, occurs much faster than this value, 1 or 2 orders of magnitude faster, in fact. So the catalytic part is far from being rate limiting here, so it's not interesting. It has no influence on kcat because it's a very rapid process. This may not be a big surprise, actually, because that process resembles very much the process. The chemistry of the cleavage is the same as the process that occurs in soluble system, and hence the rate of it is also approximately the same. So kcat here represents some other non-covalent transitions that are very slow. So kcat is simply the slowest first order step in the reaction that you're watching. And for cellulases, I've never seen the actual chemical part being rate limiting. It's always some non-covalent interactions associated with complexation or decomplexation. So that's a kcat I'm going to use henceforth. You can also, of course, define a Km, which is a half saturation concentration. And in this case, for cell 7a, it's about 5 grams per liter. So here's a table, and we can already start to do a little bit of comparative biochemistry because we have 2 enzymes, and we can see they're different here and there. But the idea here is that if we want, we can measure, well, I should say first that, so kcat we can compare directly with other enzymes because it has a usual unit per second. For Km and the specificity constant here, the ratio of kcat and Km, it's harder because they have mass units which are odd and not comparable with soluble systems, with the parameters you get from soluble systems. So, but when you measure the density of attack sites, and here are some measurements of that parameter, I'm not going to go into how you measured it. It's described in this paper here. You can, this is a kind of a conversion from mass to molar units, and then you can get the apparent molar Michaelis Menten parameters for these enzymes. And it's interesting to see, well, I think it's interesting if you compare that with normal, so to speak, normal enzymes that act in the soluble, insoluble, insoluble substrates, so in homogeneous catalysis. Here are some values. These are taken from the papers here. So these are average values in the by considering a lot of reactions of enzymes on their native substrate in the Brenda database. So these are approximate average values. And what you see is that a typical Km value for enzymes in that database is 100 micromolar. Whereas when you convert the cell 7a data into a molar concentration, cell 7a reaches half of Vmax when the concentration of attack size in the suspension is only 0.2 micromolar, 200 nanomolar. So that really talks about a very high affinity for the substrate. And therefore, actually, also the specificity constant of these enzymes, when you look at them in the usual units, is very high compared to that of other enzymes. And that's again because Km is so low. And you know, I was talking about this experimental approach to structure-functional relationships, and perhaps it's not a big surprise that cell 7a has a low Km because it has 9 subsites for a parallel subsite for binding the substrate, and hence it has a very, very tight and many interactions with the substrate. So therefore, it's natural that it has a low Km. And you can see that kind of thing in a comparative way by introducing this attack site density. Now, today I'm not going to do that very much. I'm going to stay with these parameters over here in the odd units and just say that if you use the same substrate, you can also make comparative analysis without converting into the usual units, but only if you compare all enzymes on the same substrate. So that's what I'm going to do from now on. And here's one example of this. So in this work, we took, I think, 8 different cell 6a, so this GH6 cell biohydrolases, We measured Km and Kcat in the same way as I described before for these enzymes. And you see there's quite a variety of the diversity of the kinetic parameters is pretty distinctive. If you see the blue curve here, which is the glomerular enzyme, you see that it's almost linear. So this has a very high Km and a high Kcat. Whereas other enzymes, the pink one here, which is cellulases funiculosis, cell 6A, that has a much lower Km and also a much lower Kcat. So there's some diversity in the kinetic parameters. And what we noticed in this study was that it sort of fell in different groups. So there were one groups, for instance, with these enzymes that had very high values of both Kcat and Km compared to the others. And there was another group where both parameters were very low. So we didn't find like a big distribution. The parameters always scaled with each other like this. So we thought that was interesting, and we thought that might be something that you could pursue further. So what we did, this was, of course, this was all for cell 6A. So these enzymes are pretty similar in the structure, and they also use the same mechanism, etc. So they're quite related enzymes. So therefore, we thought whether this correlation could be broadened out would be interesting to see. So what we did was we went to our freezer and we dug out all enzymes that we had that could possibly be relevant to compare in this sense. So these are all fungal cellulases, and they belong to these 5 different glycoside hydrolase families. So those are these enzymes are just about as diverse as cellulases, fungal cellulases get. So some are inverting and some are retaining, some have CBM, some don't, some are processive enzymes and some are endolytic enzymes. So they're pretty different. And then we dug out a total of 83 enzymes here and from these 5 families. And then we characterized all of them in just a simple Michaelis Menten kinetics on the same abyssal substrate. And the result of this looks like this. So this obviously is not the Michaelis Menten curve, but these are all the data compiled in one plot. So this plot lists the Kcat as a function on the ordinate here and Km on the abscissa. So what it means is that and values that are high here are enzymes that has a fast turnover, whereas those that are low and on the y-axis have a slow turnover. And similarly, the x-axis shows the low Km here to the left, which is tight binding, and vice versa, weak binding. And what we see here with these 83 enzymes is that this doesn't seem to be evenly distributed. There's a strong accumulation of data points along the diagonal of this plot. So it seems like and actually at this time we used 83 cellulases. That's a couple of years ago now that we did this work, and I think we tested another 150 since then, and they're all the same. They all accumulate on the diagonal in this plot. So that means that apparently you can't change the phenotype of a cellulase independently. There's a linkage between the turnover rate and the strength by which you bind the substrate. And to take a closer look at this, there are kind of 3 different regions here. There's a lane that's close to the diagonal, which is where almost all enzymes are, and then there's an area up here to the northwest of this fitness landscape here, where we never found an enzyme. That's a shame actually, because when you look at it, those are enzymes that has a low Km here. The Km is to the left and Kcat is high. So up here you'll have enzyme where Kcat over Km is very large. So those will be the very effective with high catalytic efficiencies. But there aren't any enzymes there, and our interpretation is that this is simply a restricted area. You can't catalyze the hydrolysis of cellulose with those kinetic parameters because of physical restrictions, and these are non-enzymatic physical restrictions. There's nothing you can't invent, or you can't evolve the enzyme structure which has these parameters because it's simply restricted by other physical limitations. There's also a wasteland of cellulases down here to the southeast of the landscape. So that we found some enzymes there. You see all these open symbols here are enzymes that have both a high Km and a low Kcat. That's a very undesirable situation, but they have very low specificity constants or very low efficacies, if you like. And we found that those were cellulases that were, those are enzymes that might not be cellulases. There are some GH12s down here that we don't know, maybe they're evolved for other substrates, There are also some that didn't, that were unstable or misfolded. One example that we looked at recently, we looked at a GH3 beta glycosidase, and to our surprise, that enzyme had a quite high activity against avicell cellulose, although the natural substrate for this enzyme is soluble celluloligosaccharides. So the kcat defined by this level here was quite high, much higher than for many cellulases, but Km was also high, so it fell in this wasteland region where the enzyme was quite inefficient, and that's simply because this enzyme has another substrate, a favorable substrate than cellulose. So our conclusion is that there is a linear scaling between kcat and Km. It's independent of the structural mechanism of the enzyme. We also see that tight binding to the substrate necessarily leads to a slow turnover and vice versa. And our interpretation is that we see this accumulation here because evolution has improved the cellulases and moved it to the left and downwards in this diagram here. But as evolution cannot overcome the physical restrictions of the reaction, then the cellulases accumulate here around the boundary for this optimal performance. We illustrated this a little bit more through some engineering, and let me just give you one example of that. So that if you take again cell 7a here, which here is the values of the wild type, it has my cursor is a little funny now. It has a pretty low value of Km. It binds very well, and also a low value of kcat. It turns over slowly. If we took a tryptophan residue that's in the binding site, which is well known to be associated with substrate binding, and replace that with alanine, we saw a huge increase in Km. So Km shifted all the way up here. I can't, for some reason it doesn't work here. Well, it shifted up here. So Km shifted halfway between the extreme values that we saw in the whole study. So just one point mutation shifted Km halfway over the whole entire diversity that we have observed. But interestingly enough, the kcat increased commensurate. So the point here did not drift into the wasteland, but it just shifted upwards along the line. So we got a different kind of cellulase, which was still on the boundary of the physical restriction. It was just a much faster and much weaker binding enzyme. And the same is true for cell 6a. And you can see similar changes by adding or removing CBMs from these enzymes. And I think I'll skip this. So for the rest of my talk, I will try to illustrate to you one applied, and if I have time, which I may not, also one basic aspect of this linear scaling. So the applied aspect is how we can use this for predicting the kinetic parameters of cellulases, and the more basic aspect is what is the physical origin of this scaling. But let's start with the applied version first. So it is quite clear, as stated here in red, that if there is a linear scaling between kcat and Km, it simplifies their prediction. Because instead of having to calculate two or compute two independent parameters, you just compute one and the other follows because they are linearly related. And so you can boil it down to just computing one. And the best way to go is to calculate the strength of binding, which gives you an apparent Km value, but by a computationally fast or cheap method. And once you have this value here, you can obviously read off the kcat because it's on. You assume that it will be on the line like the other cellulases, and that simplifies it enormously because it is very difficult to calculate. Computational determination of kcat requires quantum chemistry calculations and a lot of hassle, whereas calculating the ligand binding strength is a very well-developed and much easier problem because it's well-developed because it's extensively used for screening drugs. Right. So we tried to do it in one way, and I emphasize that you could probably do it in many others, but we tried to do this by using a so-called linear free energy, or linear interaction energy approach. So what that is it's well, I can go through it here. What we do is we start by taking the sequence of a given cellulase, and then in most cases we don't have a structure, so we do homology modeling, and we place a cellulose strand, a DP9 cellulose strand in the catalytic domain, and we place if the enzyme has a binding domain, we place that on a piece of cellulose, and then we run very short molecular dynamic simulations, only a few nanoseconds, And out of that, we get a structure, and we count the contacts in this structure, how many interactions are there, and we calibrate that against experimental data, where we had a family where we did this for ligands where we knew the affinity, and that sort of gives us an approximate value of the binding strength. Once we have that, we do what I said before. When we have the binding, we read off on the line, we read off then what is kcat, and that will give us, as a final output, predicted value of the Michaelis constants and the turnover number, or the maximal the turnover. So essentially what we do is we start with the sequence, and we end up with the kinetic parameters. And this is a very primitive way, so it's done quite fast. So on the mainframe that we have at DTU, we can handle a few hundred enzymes in a day in this way. And you can probably find other methods that can do still faster. So this is not like high throughput, but you can still cover quite a few enzymes. And it's not a precise determination of the kinetic parameters, but it's a good way to sort out if you want a certain property, you can find it in this way. So here are some screening results. This is we screened the whole GH7 family, and here's the distribution of energies that we found. So when you look at the abscissa here, you see that it's about 40 kilojoules per mole difference between the weakest and the tightest binding. And you have the well-known enzymes here. This is cell 7A from Trichoderma reesii, and cell 7B, which is an EG. So and you see here the red stripe here is the top 10 binders, and that way you can sort of learn something about the energy of the binding. You will see, for example, that here we have plotted them as where the yellow profile here is the CBHs, and whereas the purple profile is the EG. So you see CBHs bind on the average more tightly, but there's some overlap. And you can also see the effect of the CBM. It's hardly a surprise that the green profile here, which is enzymes with a CBM, binds on the average more tightly than the red profile here, which is enzymes without. But actually, there's quite a big overlap. So you can actually evolution has two ways to improve affinity. It can be both in the CD and the CBM, and there's certainly an overlap there. And here's also something where the origin of the organism. But let's just say that. Now, this was the energies that we calculated. Now we want to see, can we apply this in enzyme discovery? And the example that we are trying to establish is trying to identify a GH7 with a high catalytic efficacy, kcat over km, because we know from previous that these are good at breaking down biomass. So now we want to see, can we find previously uncharacterized enzymes that are good at, and can we predict those that are good at breaking down biomass from this approach? And to illustrate this, we organized our result in this phylogenetic tree. So you see here all this is based on the catalytic domain only, the sequence of the catalytic domain, and you see that there are two pieces of information here, except for the phylogenetic relationships, and that's the perimeter here has a color code which shows the binding strength. So red here means a very weak binding, and white is intermediate, and the brighter green, the tighter the binding. So and we quite arbitrarily cut this into different groups, and group A and B over here are the EGs, and you see group A here is EGs with no CBM. The dots in the outer perimeter here is whether the enzyme has a CBM. So here are no CBMs and very poor binding, so it's a very red. And you can go around here and find different clusters of binding strength. And we were interested in tight binding, so we chose some test enzymes. And so we wanted to go in the bright green areas, and you see here group E and F and H over here are the ones, not G, but H over here, are the ones that have the brightest green, so they have the tightest binding. So we selected 3 enzymes, one from group E, one from group F, and one from group H. They are all CBHs, and they are previously uncharacterized, and we expressed these enzymes in Aspergillus oryzae, and we characterized them. So this is how it looks. Here's the computational prediction. So this is the same distribution of binding energies as I talked about before. And you see these colored arrows here are the 3 test enzymes, the leads that we did. And you'll see that the prediction said that the one from group E was a particularly tight binding. It's way out here to the left, and the other ones are very close and a little stronger binding than the enzyme from Trichoderma. That's what the prediction said. So then we characterize them on Navicell the same way as all the other enzymes. And what you'll see here is that, again, the yellow mark here, which is the one from group E, actually confirmed that this was the strongest binding. You see, this is Km, the same way I plotted before. So this enzyme here has the lowest Km. In fact, it's the lowest Km we ever saw. It also has the lowest Kcat. It's a very, very slow enzyme. And that was, of course, predictable out of the linear scaling. The other two enzymes here are actually binds less than cell 7A. You see, they should have bound more according to the prediction, but they didn't. So it's not a precise determination, but it gives you a hint of where what type of enzyme you're, what kind of phenotype you're facing. We also tested them on biomass, and this is the result of that. So you see again, this enzyme from the group E, which has a tight binding, also has a very good, especially here after 24 hours. This is the conversion of, so this is a degree of glycan conversion of pre-treated cornstarch. This is a simple enzyme cocktail consisting of the CBH plus the BG. And you see that this is actually the best of the enzymes that we have tested here, whereas the other ones that have lower affinity are less effective. So the conclusion here is that we can kind of reasonably predict the kinetic parameters in this way. We see that the predicted tight binder from group E outperformed other enzymes that we had to our disposal, and also maybe as an interesting and a little counterintuitive result here, we see that here the enzymes are listed according to their binding affinity. So these enzymes have a low Km, and these enzymes out here have high Km. And you see the best ones are the ones with a low Km. So they are also the enzymes with the lowest Kcat. That follows from this line that I've been talking up and down about now. Low Km also means low Kcat. So actually, the best indicator of a good biomass degrading enzyme is that it's a slow enzyme. It has to have a low Kcat, then it's a really good enzyme against biomass. That could be, that I think is counterintuitive, but it's simply because affinity is more, the tight binding is more important than rapid turnover. And I see Morten on my picture now. I know that's a sign that I'm running out of time. So I'll skip the physics of the origin of this scaling and just go to my closing remarks. So in closing, if you consider a parameter we call the attack site density, so that's how many moles of attack sites there is in 1 gram of cellulose, you have a basis for doing simple steady-state kinetics on insoluble substrates. You get a situation which it's like these two tomatoes here. It's the same but different. It's very similar to conventional Michaelis Menten kinetics, but just with a little flavor, different flavor and one extra parameter. One result of using this is that Kcat and Km for cellulases cannot be varied independently. They scale along this line here, so they are limited by a so-called linear free energy relationship, which is something that is very commonly seen in organic catalysis. And it simply has to do with that the tighter you bind, the bigger the barrier for transition. But that was what I had to skip, so I better skip that here also. This means that the kinetic diversity of cellulases is mainly a matter of how tightly they bind the substrate. That's the main descriptor of the different kinetics that we measure. Almost the entire difference lies there. And finally, and perhaps more interestingly, this linkage simplifies computational predictions, and you can use that to discover enzymes, both wild types, and you can also predict the kinetics of the artificial variants that you make for technical use. So that just leaves me to thank you very much for your attention, and I'll just show you my acknowledgement. All these wonderful people are the ones that have made this work, and these different icons to the right are the foundations that have supported us. Thanks very much.
Thank you, Peter, for a really, really interesting and nice talk. We now open the floor for discussions or questions. Bernard, do you have a question? You can speak out. I think you should be able to talk. Not, I can just say your treatment of tech. Okay, Bernard, come on.
Yeah, well, great talk, Peter. and the treatment of the attack sites in the theory is really great. Did you consider that all attack sites are equal, or could you represent the attack sites as an ensemble of attack sites of different reactivity and different accessibility? In other words, is it not just oversimplified, and could we perhaps be more precise in the treatment? For instance, we all know that Avicel is pretty heterogeneous in terms of accessibility. There are some areas that will be obviously more accessible than others. And what do you think of that?
That's an excellent question, Bernard. Thanks for that. It is a simplification. And right in this treatment, I just hope that it's okay to consider it as an ensemble where I sort of look at the average. That's not right, actually, because since we did this, we started to introduce distributions of attack sites. That there are good and bad attack sites. There are some that are easy to get for the enzyme and some that are much harder to get. And you will get different. You will get a different interpretation. Not the K-cat is actually unaffected. That's just a simple average. But for Km, you get a different interpretation. So that will change this. So it's an excellent question. But I can't help saying that this is a situation that resembles very much what happened 10 or 20 years ago when single molecule enzymology was launched. And one of the big results in those days were that that the enzymes, that K-cat is not the same for every enzyme, because there are slow structural fluctuations in the enzyme. So sometimes the enzyme is in a good position and it has a quick K-cat, and sometimes it's in a lousy position and K-cat is slow. So actually, I think the same type of heterogeneity considerations that we are definitely have to take into account to describe these heterogeneous systems well is actually also known in the bulk phase.
Yeah, well, I could not agree more. And also, obviously, the concentration in attack sites will rapidly evolve along the reaction pathway. Yes. And I was just wondering whether your kinetics there, and you showed that in a certain in certain cases they are, whether they reflect the actual performance of the enzyme in a reactor rather than in a test tube. And I think you have shown that indeed it is the case. So congratulations. Yeah,
there is some, but you're absolutely right. Again, it's not always the case. We often see that you know, that even small changes in the condition, let alone going from a test tube to a big reactor, changes the ranking of the enzymes that we are looking at. So it is a very relevant. It's a very relevant consideration.
Thank you.
I have a question from Morten Torborg, Novozymes. Your best enzymes are the slow ones. Would that be the case if your slope was really 1 and not less than 1?
No, that's a good question. No, I don't think so. It's because the slope is less than 1. So that means, when we plot this, you see that, just to make that we're on the same page, right? We have K-cat on one axis and Km on the other. So the slope has to do with the specificity constant. And if the slope was 1, it would mean that every cellulase had the same specificity constant. And then I think it wouldn't matter. But since the slope is less than 1, then it means that the specificity constant or the catalytic efficacy increases when you go to lower Km values, because the slope is not 1. So this is a very relevant point. That had the slope been 1, then they would all have had exactly the same specificity constant, but they don't. The higher specificity constants are the enzymes with the tighter binding and the slower turnover.
And then a question from Vincent.
Yeah, hi, Peter. Very nice. I have a question that I'm sure Morten could also have asked. I was just wondering why the word processivity was not used in your talk. I mean, how do you deal with that in your kinetic assessments? And you may be aware of some work we have been doing on gytinases where we're looking more at processivity in connection to similar things.
And it's very related because, you know, processivity again has to do with the binding strength, right? Because in the simplest interpretation, it's just a statistical thing. How often do you fall off, and how often do you take another step along the same polymer? So those things are very related. So I remember your paper. Was it 10 years ago or something about that? And I think it's the same. It's the same thing we're seeing. But returning to your first question, that's another. I have to just to admit that yes, in the way I talked about it today, I totally neglected processivity, which is a you know, and probably an unacceptable simplification. But that's the best I could do at the time, because so the attack side is a strange thing, right? So for a cell biohydrolase, the attack side is getting started and then running down the track, so to speak. So the attack side is that's all the sites that it can hydrolyze, but they're kind of different because that's the starting point and then the running points. So therefore, and I neglect that totally and just say, okay, we pool everything together and say they're just the average of everything. So it's a very, very, very primitive description I presented, and we are trying now to introduce processivity into this, but the problem is, as you know very well, that processivity is very difficult to measure precisely. So when you try to account for processivity, very often you drown your model in uncertainty because you get very big uncertainties from the processivity you measure. Even the EGs have quite high processivities actually. So, and they're very hard to measure. So we would love to do something more along those lines, but this is just a very coarse way to do it so far.
Thanks. Very nice.
I have just one quick question to end this this talk here. It was really interesting to see that when you took off a tryptophan or a CBM, you just hopped along the line, you stayed on the line.
Yes.
On the early slides there. Is this because dissociation of the cellulose is the rate determining step?
Yes.
I mean,
it's related to that. It's related, I think, to two things. One, the first requirement is what you say. It's because the dissociation is rate limiting, at least it determines kcat. So when there's a lot of substrate, dissociation is the slowest step. But it's also a sign, I think, that the physical restrictions I was talking about, the non-enzymatic limitations of the process, are very severe. That means that you could say that it's very easy for evolution to hit the boundary. So that means that even if when we take cell 7A and we remove the most conserved residue in the whole enzyme, then we're still on the boundary. That's a surprise because you would have thought that you mutilated the enzyme that got a very poor catalytic performance, but you're still on the physical, on the boundary of what's physically possible. So I see that as a sign that the physical constraints are very severe, so that you can say that you have to really mess up your enzyme before it actually falls off the line. And we can do that. If you take out the nucleophile, then you fall off the line, right? But you have to really torture your enzyme before it starts to move away from the line, because the line is a severe restriction.
Well, thank you, Peter. When we meet in June on Norwegian next year, I'll talk to you a little bit what kind of consequences that we have for GH18s, where the rate association, how much association is the sort of the kcat rate limiting step, and how that would be. So, well,
yeah,
thank you so much.
Thanks, Martin.